In the previous video, we discussed about the diabetes mellitus. If you want to watch that video first, the link is in the description. Now in this video, we'll be discussing about the type 1 diabetes mellitus in detail. First of all, we see it's the autoimmune disease, where our immune system launches attack on our own cells, like we have our T cells attacking our pancreatic beta cells. So that way, it's a type of type 4 hypersensitivity, where our immune system has lost the self-tolerance, thereby attacking their own cells. If we see its genetics, this autoimmune disease is mostly associated with HLA genes, that's human leukocyte antigen genes, which includes two specific proteins for type 1 diabetes, that's HLA-D3 and HLA-D4. So when we have this autoimmune disease, we get the generation of autoantibodies. Mostly we get three important antibodies. First one is the anti-islet cell antibody, second one is the anti-GAD antibody, and third one is the anti-ZNT8 antibody. So these antibodies attack the beta cells of pancreas. That leads to less or no insulin production at all within our body, which ultimately leads to accumulation of glucose within our blood. As we know, this insulin opens the gateway for glucose to get into our cells. That mechanism we are going to see later on in this video. Now let's get to the mechanism of type 1 diabetes. Here in this diagram we have the pancreatic beta cell. It has got GLUT4 channels for glucose intake. And on the left it has got ATP sensitive potassium channels for potassium transport out of the cell. Then we have calcium channels, and we also have the insulin vesicle, which has got proteins on its membrane in the form of islet antigen 2 protein and ZNT8 protein as shown in the diagram. So in order to start a pathway, first of all the glucose molecules gets into the cell as shown in the animation through GLUT4 channels. And within the cell, the glucose is driven into aerobic respiration through glycolysis, citric acid cycle and oxidative phosphorylation. And eventually we get the ATP generation. And when we accumulate lot of ATPs, these ATPs act on ATP sensitive potassium channels and inhibits them. So it must be noted that these ATP molecules inhibit the potassium channels. That leads to accumulation of potassium ions within the cell as shown in the animation, thus causing the membrane potential of the cell to depolarize. And with this depolarization, we get the activation of voltage-gated calcium channels as shown in the animation. So here we see the calcium channels are getting opened, and with this calcium ions flow into the cell through these channels. And these calcium ions drives the exocytosis of insulin vesicle, thus promoting the calcium-dependent release of insulin as shown in the animation. On the other hand, we also have glutamate molecules within the cell. These glutamate molecules are acted upon by GAD enzyme and converts it into GABA. And this GABA also mediates the release of insulin from vesicles. Now when we have the autoimmune disease, which has unknown causes till now, but in some cases we have the presence of HLA-D3, HLA-D4 proteins in type 1 patients. But it must be noted that not all affected ones have this HLA genetic abnormality. So under these circumstances, we get the production of autoantibodies with anti-islet cell antibody, anti-GAD antibodies and anti-ZNT8 antibodies. The first antibody, that's the anti-islet antibodies, comes in and targets the islet antigen 2 protein on the insulin vesicle. This way the insulin vesicle exocytosis is affected. The second antibody is the ZNT8 antibody. It comes in and targets the ZNT8 protein on insulin vesicle and affects the secretion of insulin also. And finally we have the anti-GAD antibody. It targets the GAD enzyme and halts the conversion of glutamate to GABA. So that way GABA mediated insulin secretion gets affected. Furthermore, when we do not have enough insulin, that means insulin mediated GLUT channel expression in the cells will be affected, where our cells do not have enough GLUT4 channels for glucose intake. So that way when insulin is absent, the glucose will not enter into the cells for ATP generation. So by this way we get the accumulation of glucose in the blood, or we can say high glucose levels in the blood, which is termed as type 1 diabetes mellitus. Now let's have a look at major symptoms of type 1 diabetes. We know glucose accumulates in the blood. There is no ATP generation or less ATP generation. That leads to lipolysis or protein breakdown. So due to all these factors, first we get the polyphagia, where a person gets into excessive eating under the influence of hyperglycemia. Then we have glycosuria, where an affected person passes excessive glucose in its urine. Then we have polyuria, which simply means excessive urination. And when affected person has excessive urination, that leads to polydipsia, which means excessive thirst. And do remember, these are the 3 P's which are commonly attached with the diabetes mellitus. That's polyphagia, polyuria and polydipsia. So this is what type 1 diabetes is and the mechanism behind it. I hope you like the video. If you like it, give it a thumbs up. Do consider support my work on Patreon or YouTube and make sure to subscribe this channel. Thanks.